Hi, everybody. Welcome to today's seminar on rare diseases and human genetics, and this goal that the Human Cell Atlas Initiative has to try and contribute to all communities of research in human genetic diseases. And so we're on behalf of the developmental biology group of the Human Cell Atlas, welcome all the speakers and panelists. We have a fantastic group of speakers and panelists for today, and then we're going to have a sister meeting in next week for Asia and Australia, and also some fantastic speakers for that. And so putting the two meetings together, what we're trying to catalyze here is lots of thought and initiative and smarter experiments and smarter data analyses that can inform mechanisms by which genetic mutations cause disease. And we have Heidi Rehm to start us off talking about human genetics and pathologic mutations. She's been just a tremendous leader in this area, and you know, it's not only talk the talk, she's walked the walk in developing standards and contributing large amounts of data to ClinVar, and thinking about the next versions of databases beyond ClinVar that we'll really need in order to solve problems at scale. So, and then we'll have Jay Shendure, who's doing some absolutely spectacular work in developing human fetuses and profiling different cell trajectories, and we'll see genes in action. So, and then we have a fantastic panel too, Heather Etchevers and Ewan Birney and David Fitzpatrick, all just tremendous contributors to our understanding of how gene networks allow for health, and when they go bad, cause disease. So with no further ado, Heidi.
Thank you. It's a great pleasure to join the HCA for this session today on rare disease and applications. So I'm going to talk about exactly that. A little more focus on how we advance rare disease studies through different types of collaboration. Let's see, get started here. It's advancing. There, oops, there we go. It's a little slow. So this is our team that Anna Donaluria and myself co-lead, that's really been focused on our rare disease gene discovery efforts, both through our NIH-funded Center for Mendelian Genomics and then another project called the Rare Genomes Project that I'll mention briefly. And you know, in our rare disease studies, we focus a lot on scaling rare disease gene discovery through automation, producing software that the community can use and collaborate on, like our Seeker platform, doing methods development, both ourselves and with collaborators like the Taukowski Lab in structural variation, working with RNA-seq, et cetera, and trying to expand cohorts to enable us to shift from just family-centric studies to cohort-based analyses. We continue to use exomes as our workhorse, but we're increasingly using whole genome and combined with RNA-seq for looking at exome negative cases. And we do also rely really heavily on data sharing, both to assist ourselves as well as to engage the broader community in making advances. And I'll talk about a few of these different platforms that we're using. We work with a large network of collaborators, in fact, over 44 collaborators from around the world, many of whom are shown here, collecting samples that have been recruited from many different disease-specific studies and broader studies. And to date, because of this large collaborative network, as opposed to just working, you know, ourselves in silos, we have been able to discover lots of different gene disease discoveries with this network. To date, our stats are 821 novel gene discoveries or candidates that have been generated to date through our 4 years working in this area. That includes 181 that are the tier 1. These are well supported, either already published now or on their way to publication, novel gene discoveries, as well as 91 phenotype expansions. So the gene's already been implicated in one disease, but we've now implicated it in another phenotype. And then another 550 or so that are candidates really needing a little more evidence to really prove their causality. And that's work that's ongoing across ourselves and all of our collaborators. We also launched the Rare Genomes Project as a way for us to directly recruit our own participants. That allowed us to be in full control of the data sharing approaches that we take. And so we actually share data with no embargo. The moment we generate it, even if we haven't looked at it yet, we put it out for sharing with the community. And we also are able to return results directly to those participants through an IRB approved process. This allows us to engage participants in this process, and we return both clear causes of their disease, and we also return very suspicious candidates because we find they can engage and help us with the further steps in validating those discoveries. For the Rare Genomes Project specifically, we've now enrolled over 500 families and returned results to 74 of those families and are actively pursuing a number of candidates. This is often done collaboratively with people around the world who are studying those specific genes that we can identify. So we have 21 functional studies in progress for many of these candidates, 18 publications in progress across a lot of different experimental systems that can help us validate these candidates that we generate through our sequencing studies and analysis studies. We did look specifically as we continued to wrestle with, should we be doing genome versus exome as we think about cost and yield. So this study, Anne and Monica were both looking deeply at our data from the first 500 families that we did genome sequencing on. And if we focus on the 128 that were solved or likely solved, looking at which ones really required genome sequencing versus exome. So about 6.7% did require the genome for analysis. A lot of those were for structural variants, as well as about a third for missed coding variants where they were not well covered in exome sequencing, and then about 8% for non-coding variants that weren't targeted at all in exome, and one short tandem repeat. Some of the things that were solved by exome but weren't recognized at first was because of subsequent discoveries of genes after those cases were analyzed, or adding RNA-seq data to be able to better interpret things like that. So in terms of once we identify candidates, this is where collaboration really becomes critical. And I love this image, this analogy that Kim Boycott put together, stole this slide from her, where we think about different sided matching in the rare disease space. So two-sided matching is what we do in the Matchmaker Exchange. And we launched this platform. Now it's been over 5 years ago since we launched this, and it's a really great example of federated data sharing. So the 7 major nodes of this platform exist in different countries around the world, where their data can stay in the place that they need to be protected, but then we can, through application programming interfaces, query back and forth and say, gee, I have this candidate gene. Do you have a candidate gene from your studies as well? And it's kind of like go fish on a global scale. And a lot of the infrastructure to support this was done in collaboration with the Global Alliance for Genomics and Health, which is really a standards building organization for genomics and health, where a lot of the APIs that were needed to exchange data across this platform and figure out how we authenticate and allow permissions to share data and structure that data was developed in collaboration with the Global Alliance as a really critical process. So this platform is now used pretty much by every rare disease group around the world. It's just incredibly powerful. There's over, data from over 150,000 patients from over 4,000 contributors from over 84 countries around the world. This is the data from each of the individual nodes that's in there. And I would say in practice, nearly every gene discovery we make, we use Matchmaker Exchange in some way, shape, or form. Sometimes it's that first match we get that starts the process of discovery. Other times we throw in a candidate and discover that there's already a whole group of people that have already matched, and we can add our case to their publication that's just about to submit. So it kind of runs the gamut of the stages of gene discovery in the matching that is done in this platform. However, if you look at the left side of the slide I showed, there's still a huge percentage of cases in all of the groups doing rare disease discovery that are still unsolved, despite our best efforts. So how do we continue to tackle these? Well, if we go back to the paradigms of data sharing, next is what we call one-sided matching. In this case, one group may have a candidate, and you're looking to match, but you put it in a matchmaker exchange, and there are no matches. But somewhere out there, there's some case or many cases that might have a variant in that same candidate gene. So could you query the raw data and look for that candidate that's sitting that never got recognized? And so there's now platforms developing for this. A couple of that are out there are Variant Matcher from the Hopkins group. And if you put in your variant and include your phenotype, like I did at the top here, they will return specific matches with the detailed phenotypic data, which is shown here in the rest of this right side of the panel, that I got back through email doing this specific match where they had not identified this variant as a candidate, but I was able to query their raw data and get back the detailed phenotype for all the cases with the variant. We also put all our data into Geno2MP, a platform supported by the University of Washington, CMG, and that allows it to be queried by many collaborators around the world that are querying that database for matches. And we get routine emails, with matches. So a couple of one-sided platforms. Of course, sometimes we have data that no one can figure out any candidate in, but if we bring those data sets together, we can try to bring signal out of the noise of all the variation in these data sets. We call that zero-sided matching. That's where platforms like Anvil, that's being supported by NHGRI, and other large-scale platforms for genomic data sharing become really critical. And so all the CMGs in the US are now putting all of their data into Anvil. Ours is all in there already. And not only is the data there, but richer annotations and metadata are there so this data can actually be used productively by the community in terms of affected status, family relationships, candidate variants that have already been solved, all those annotations there. And in fact, after we loaded up, a round of data, a colleague in Utah was testing out a structural variant caller and was able to solve one of our Rare Genomes Project cases before we'd even started analyzing it with a structural variant. So really great ability to rapidly share data and allow engagement of the global community in solving cases. Another platform that's being built in the US is the All of Us research program that's now enrolled close to 500,000 participants, really targeting underrepresented populations, both racial and other demographics. And this data is now, being put into a data research center along with electronic health records, participant provided information, physical data, biospecimens, other information that will make this a very rich resource for us to use in many different studies. And although it's not specifically recruiting rare disease patients, there will be across the million subjects, participants that have rare disease, given that on average about 1 in 10 people have rare disease in our population. And that data is being apportioned into public, registered, and controlled access and allowing a really wide net of people that can access this data for studies. And we are a funded genome center for the All of Us program. And so we've already started genome sequencing. The goal, I think it moves daily, but to probably generate over 100,000 whole genomes worth of data throughout 2021, as well as 300,000 arrays on the data. Probably every participant that will have been enrolled next year will have arrays as well. And we are working to return results to participants and also joint call that data so it can be included in the NOMAD data set, which is critical for all of us in terms of population studies. For the return of results, this is where variant interpretation becomes critical. And there are 4 clinical labs participating in the interpretation of variation from the All of Us research project to be able to facilitate return of results. So to prep for that, this is an FDA process that we had to go through in order to get permission and have approval with an IDE to return results to participants. That required us to harmonize across all 4 labs so that our interpretation of variation is identical. No result can be different between the 2 of the different labs. So when we compared 50,000 sequence variants from the first set of genes we will return to participants, the ACMG 59, we were actually 99.1% concordant across these 50,000 sequence variants, or the 24% that had at least 2 interpretations. So that's actually demonstrates that our community has come a long way in terms of improving standards for variant interpretation. Now, we still had 100 or so we had to resolve, and we went through rounds of resolution. We're down to 12 that are part of the pre-harmonization. We will also, once we launch the return of results that will happen in 2021 to participants, we'll have to real-time harmonize so that no result is different. And when we can't resolve between labs, we will push these to the ClinGen expert panels, which brings me to that project, which is, as Bruce mentioned, I spent a lot of time in developing standards and resources to support genomic interpretation, both variant level and gene level. And this is a large NIH funded program with over 1,500 people around the world participating in our expert panels to curate knowledge to support both rare disease studies, and we also are now supporting cancer interpretation. And we do that through a number of different approaches. On the variant side, we develop common standards. We facilitate public sharing of interpretations through ClinVar. We facilitate inter-lab conflict resolution between the labs, and I just showed an example of that with the All of Us program. And then we engage experts for all those variants that can't get resolved through these other approaches. And we have over 12,000 expert reviewed variants in ClinVar. On the other side is the gene curation. So as many of you know, lots of groups publish papers that claim new gene disease discoveries. We do that ourselves. ClinGen developed a more standardized framework to evaluate the validity of that evidence for gene disease relationships, looking at both genetic and experimental evidence, and then scoring that on this sort of set of tiers here, definitive down to refuted data. And we've evaluated now 1,134 gene disease pairs through all of our different, over 50 different expert panels working in their specific domains. And what we find fairly commonly is about a third of those genes that have been reported as associated with disease don't have sufficient evidence for one of those claims. So this is a challenge to our field and a need to continue to push in building evidence. We do find that most of the genes that have limited evidence are because there was just 1 or 2 cases that were identified with candidates, and then the functional study was what was reliant, and those functional studies aren't always optimally designed to recapitulate the human disease and aren't always valid ways to implicate the gene. And that's been one of the big challenges that we've seen in the field. All of these curations are downloadable off the ClinGen website, either in aggregate or you can browse for your favorite gene. Now we have a larger project or an extended project called GenCC for the Gene Curation Coalition. This brings together not just ClinGen, but many other groups that work in gene disease validity curation. That includes some public resources like OMIM and PanelApp and the TGMI Gene to Phenotype group. David's on the call today who works on that. And also resources that aren't public from the clinical labs that have been curating genes. And we have been working with these groups over the last year or two to harmonize efforts and bring terminology together. The next slide. We had to take all different terminologies groups were using for classifying gene disease relationships, and through a Delphi survey process, harmonize those terms. So we would all be using the same terms, which are very similar to the ClinGen set. And in the next week or so, we'll be launching the GenCC database that will have all of this data, thousands of gene curations from lots of different resources, all brought together. And so you'll be able to type in your favorite gene, which is just shown here in the screens that we'll be launching next week. And you can search for a given gene and look across and see how many claims that are definitive or moderate or refuted for each of those genes. You can then click into it and see the detail. For what condition was it definitive or refuted? Who made the claim? Links to those databases for the supporting evidence, or sometimes it's directly submitted into the database if there's no public website for it. And you'll be able to get all this information from all these different groups in one place, download the data and use it to your heart's content. So we're really excited to launch that. And so watch Twitter, it'll be coming out soon. And then the last bit I'll talk about is Nomad. And so we just had a release at the end of October, It didn't have a large number of new genomic data sets, although it had some critical ones for diversity, including the Human Genome Diversity Panel and the 1,000 Genomes data. We now have additional ancestry annotations in there, including Middle Eastern, which we never had before. All of the Nomad data is now freely available from all 3 cloud service providers. We now have read supported data for all of the non-coding variants. It's over 2.5 billion genotypes. That was really a struggle to load into the system. Manual curation for homozygous predicted loss of function variants that helps figure out if these genes really are disrupted in humans or not for real. Added in silico predictors, and now we have added mitochondrial variation, which wasn't there before. So this is some screenshots from the mitochondrial variation viewer in Nomad with both homoplasmic and heteroplasmic. A lot of QC went into this through Sarah Calvo, who's in Vamsi Mutha's group. Nice collaboration with Vamsi's lab and Monko Lex lab. Daniel, others involved in this effort to give really high quality mitochondrial data to the Nomad browser. And I'll thank the core Nomad team shown here, and of course, many data contributors for which we wouldn't be able to do this project if their data wasn't shared. So we really appreciate that. And I think that is it for all I have. I appreciate all the groups that have been working with us on these projects. And we're excited about V4 that'll be coming in early 2021 that'll add all the UK Biobank data and lots of other genomes and exomes. I'm going to turn it over to Jay.
Wow, Heidi, thank you so much. That was a spectacular blast, and just so much to talk about. But for all that work and for all that infrastructure that we're going to be able to connect that data into and come in and out of it, this is just, it's an incredible resource. So much potential. Jay, Shendure is absolutely another spectacular person on the panel today, and thank you so much for agreeing to come and give a talk. Jay's been very busy doing amazing things, as he has ever since he was in Church's lab, and probably before that too. Showing it's possible to get the sequence of a complete fetus from blood samples non-invasively from mothers, and now recently publishing human developing fetal embryonic cell atlas trajectories and showing lineage differentiation. There's incredible relevance to developmental biology and how genes and gene networks and cell networks are going to work to create healthy life. So, Jay, thank you so much.
Great, thanks, and thanks for the introduction, Bruce, and great to be here and fun to be juxtaposed with Heidi here. Amazing talk, and I think will be a really nice way to intersect these 2 topics. Okay, so I'm gonna sort of setting a timer here. I'm gonna try to do a 20-minute summary of some recent work that we published on cell atlases of both gene expression and chromatin accessibility during fetal stages of human development. And just to do some acknowledgements up front here, if that's okay. This has been work from a lot of people over a couple of years, but the folks shown on this slide, I think, have been really the principal lifters of this. So the chromatin accessibility work was led by Sylvia Damke, Andrew Hill, Riza Daza, and Darren Kusanovich in my group, and the gene expression work by Jun Cao, who's now an assistant professor at Rockefeller. All of this has been in collaboration with Cole Trapnell, as well as Frank Steemers and their advanced research group at Illumina. And then last but not least, this has been critically enabled by the UW Birth Defects Research Lab, led by Ian Glass, Dan Doherty, with huge amounts of work performed by Diana O'Day to really make this possible. Okay, so broad background here. So our group is a tech dev group, and for years has been working on new single cell methods, primarily focused on this paradigm of combinatorial indexing, which Darren Kusanovich and Riza Daza first described around profiling chromatin accessibility in 2015. And this is, not the way most people do single cell profiling, but I think some of the advantages, if you're interested in this include exponential scalability, as you'll hear about in this talk, with linear amounts of increasing work, as well as the breadth of different aspects of molecular biology that we've been able to profile at single cell resolution with this basic workflow or paradigm. So, and I'm not going to talk about how this works. It's in the literature now, and I think probably many of you are familiar with it. We have, in the course of trying to find venues in which to demonstrate these technologies, have been really focused on development, and in particular trying to do kind of organism scale atlases in a variety of model organisms, as well as you'll hear about today, the human. So this started out with worms. We've also done atlases in the fly and the mouse, and some of that will come up today. And in particular, the kind of the launching point for our studies of human development, leveraging our relationship with the UW Birth Defects Research Lab to profile a somewhat later period in development than we have previously done with focusing on mouse organogenesis. And I'll come back to this again a bit later. So the two atlases which just came out, and again, kind of can't do these justice in a short talk, but one was focused on fetal gene expression. So with this 3-level combinatorial indexing, highlighting kind of the exponential nature of the technologies, we were able to profile on the order of 4 million cells. So shown in the middle here is a kind of a global UMAP based on subsampling from different cell types. These derive from 15 human organs, fetal organs. Some are missing in their particular, I think, skin, bone marrow, and gonads, but we were able to get these from more than one individual per organ and then subject them to this 3-level combinatorial indexing that Joan had previously developed. I think one thing that merits mentioning is that these are very large numbers of cells profiled. These were not experiments conducted by large teams. The preparation of tissues was done by a single individual, and the actual experiments were done by one other individual, right? So it's just relatively small teams are able to perform these kinds of experiments because of the exponential nature of the technology. Chromatin accessibility. So this is building off of methods that we've previously reported, going back to 2015. And one thing that's new here relative to earlier iterations of this SkyAttack-seq technology is that Riza Daza and Darren Kasanovich developed a 3-level version of this, which we're put the protocol out for the first time. And in particular, I think one of the frustrations with this method has been the reliance on these custom indexed transposases, which can be challenging to manufacture on your own. But here with this new 3-level version, it's purely relying on kind of the standard TN5 product, still as Nextera from Illumina, to enable not just 2 but now 3 rounds of indexing with 2 rounds of ligation. And so with this method, again, focused on the same 15 fetal tissues, Sylvia, Darren, Andrew, and Riza were able to profile on the order of 800,000 single cells. One point of note is this is not a co-assay, so we're not profiling gene expression and chromatin accessibility in the same cells, but rather obtaining those from different cells. However, the nuclear preps from which we're performing these experiments are often the same or overlapping. So the 2 data sets do speak to each other at some level. Okay, I'm assuming I haven't heard anything, so I am advancing, I think. Okay, so one topic I'll try and touch on in a few slides here is how we approach cell type annotation. So this was largely driven by the gene expression data, which was far more powerful for discriminating and facilitating annotation, including of small clusters. And so at the highest level, I think we have, there's nothing really special about how we approach this. It's kind of in the usual way, using Monocle 3, which implements UMAP and Louvain clustering to pull out big clusters, and then kind of manually annotating these based on gene expression and markers vis-a-vis the literature and other atlases. So shown here on the left here are kind of the individual atlases for each of the 15 organs. And then we're intersecting these with one another to consolidate down to 77, quote, major cell types, at least some of which are shared across the organs, but certainly not all of which. And then to kind of bring this to another level of granularity, we of course tried subclustering, right? And so one of the challenges here, as I'm sure anyone who's done this can speak to, is you never really know when you're done, right? You can kind of subcluster ad nauseum or ad infinitum, and really trying to figure out what a good stopping point is, what subclusters are meaningful, that kind of thing is a challenge. So here's the approach that we took, which I think is, at least I found quite useful. And this was worked out by Jun Cao. So here we're taking a particular major cluster from the kidney and subclustering it. It's critical, even with these data that are performed with the uniform protocol, to do batch correction. Here we're using the harmony method from Korsunski et al. And then once we actually have subclusters and we are looking at them, we apply this method from Abidal et al. Reported in Genome Biology last year, which tries to basically take each cell and ask if it can reclassify it into one of these 10 clusters. And so this is a very neat case where the 10 clusters are kind of, you can accurately replace cells into each of those subclusters with a lot of confidence. However, it's sometimes the case that you have two subclusters that are difficult to distinguish from one another when you try this cross-validation exercise. And in those cases, we merge the clusters. And then, okay, so that process or procedure resulted in 657 subtypes that that were transcriptionally distinct through this cross-validation approach. And then we go about annotating these. And here, I think this kind of speaks to the field as a whole, where we're benefiting incredibly from extensive annotation work done by other groups, most of which is in papers really just from the last 1 or 2 years, and largely deriving from this community. So here in particular, a lot of what we relied on was the mouse cell atlas from Guo et al. From 2 years ago, as well as the mouse brain cell atlas from Sten Linderson and colleagues, and applying a cell type cross-matching procedure that Jun developed to be able to kind of provide at least initial annotations for these cell types. And I think this is really the future, where one is not reinventing the wheel with annotation every time, but even at the subcluster level of granularity, trying to recycle the hard work of others into these annotations. And so here on the left is just showing it for that metanephric set of metanephric subclusters that I had on the last slide, cross-matching with the MCA. And then here on the right is we were able to preliminarily annotate about half of our cerebellar and cerebral clusters based on cross-matching to adult mouse brain from Sten Linderson's group, often but not always, with these one-to-one relationships. Okay, and then chromatin accessibility, as I alluded to earlier, can be quite a challenge to annotate. And so here we're really leveraging the RNA data and kind of similar cross-matching procedures. So at the first level, we're performing kind of a similar clustering exercise, shown on the left here, kind of 15 organ-specific chromatin accessibility atlases. And then we perform a cross-matching procedure, do some live animation here, a cross-matching procedure with the RNA data from the same organ. And similarly, this kind of NNLS approach leads often to these one-to-one matches, not always, but often. And then that leads to kind of a liftover of these labels from the RNA clusters to the attack clusters, which we then follow up with manual visualization of promoter accessibility for key marker genes of the RNA clusters. And so showing just one example here to verify those labels. And so here's, this is the chromatin accessibility equivalent of a marker gene expression plot, right? Where here we're visualizing marker gene promoter accessibility across, these assignments that we made, and you kind of see this diagonal stripe kind of indicating a lot of these validations, right? Okay. So next couple slides, I'll just give some anecdotes from the paper around integration across species, time, and tissue. So again, just I think this also highlights the importance and the opportunity and the possibilities that of this kind of integration between data sets, even when those data sets cross not only these kinds of boundaries, but also boundaries of technology platforms. And so here's one example. So one of the principal rationales for doing a multi-tissue study in the first place as kind of a single study is the ability to look at cell types that appear in multiple tissues, right? And so amongst those are, of course, blood cells. So here we're just pulling out the blood cells and clustering them independently of everything else. So colored on the left by organ and colored on the right by annotated cell types, right? And so this is, you can see that at least in some cases, or in most cases, at least for blood, as you might expect, we don't see a ton of organ specificity for different cell types, right? And this contrasts with, say, endothelial cells, where there's quite a bit of cell type specific gene expression programs that are restricted by organ. And then now here, just showing kind of leveraging other data sets. So here we've integrated the data in our study, which is at a relatively narrow window of human development, but from many different tissues, with related data from this human fetal liver atlas from Hanifa and Teichmann and colleagues last year. Really beautiful study, and we're able to overcome kind of the technical challenges. Again, kind of relying on tools like Seurat and others for this kind of data integration exercise. And when we bring these data together, what we find is that those gaps between different clusters of blood cells we can bridge through their common origin at earlier stages of fetal development, as viewed through the Popescu et al. Data. And really, the data sets are kind of mutually beneficial, I would say. We're not able to draw these connections without these earlier time points, but by having multiple tissues beyond the liver, we bring to the table kind of the immune cells that are in other organs and not necessarily sampled if you're just focusing on the liver. And so you can pull it together to build kind of this more continuous set of trajectories in space and time. Okay, so extending that a little further to bringing together the mouse and the human. So one of the things that surprised me the most in all of these kinds of analyses is just the ease with which we can, again, I shouldn't say ease because it does rely on a lot of really impressive tools to be able to do this successfully, but to bring together, let's say, mouse and human development. So this is such an integrated view. So I mentioned earlier that we had profiled mouse development between E9.5 and E13.5, and in humans, we're profiling this somewhat later stage of gestation, of development, of mammalian development. And so facilitated by Seurat, we're able to integrate these things. And so sort of the progression from E9.5 to E13.5 that we saw in the mouse data kind of continues directly into human fetal development, and this representation shown here, where the dominant signal here is not the species differences, but rather kind of the differences between major trajectories of development. Okay, so just one more example of that, of showing how this can be useful. So in our, one of the challenges when you're studying early development in a mouse is that you can't necessarily dissect out every organ, right, or every tissue. And so here we're actually benefiting from the anatomical resolution of the human data to some degree, and integrating that back with the mouse data. So here, these are mouse, kind of one big set of macrophage and microglial clusters that we had all kind of lumped together in the original study. And then when we use that together, or we co-embed that with the human fetal data, we're able to actually separate out different subsets of mouse cells, including different classes of macrophages and microglia from one another, as well as to look backwards in time to early programs where those separate from one another. Okay, I hope I could get a couple extra minutes just because the technical snafus. I know I'm pushing up against 20 here, but just a couple more
You are,
slides.
but go for it. This is so utterly delicious.
Okay,
But
so
keep rocking.
how can we leverage gene expression and chromatin accessibility together, since we did these again on kind of the same tissues? So if we look at just chromatin accessibility alone, we can of course define large sets of large numbers of regulatory motifs that are enriched for chromatin accessibility in cell type specific ways. But we can also think about how to intersect with gene expression. So in particular, we can look for correlations between gene expression and chromatin accessibility of the cognate motifs of those transcription factors. So the example shown on here on the left, this is looking across different individuals and cell types across our entire data set, where we see positive correlation between the expression of the TF on the y-axis, SPI1, and the accessibility of its cognate motif. So that implies that this is an activator, this has a role in opening up chromatin. On the other hand, on the left, we have an anti-correlation where the expression level of this particular motif is correlated with closing of the cognate of the cognate motif, or regions with the cognate motif in chromatin. And it's important to note that this is not, it's not always the case that a repressor is associated with closing of chromatin, right? So there are some repressors that are known transcriptional repressors, so like solid cases, but you actually see it's associated with an opening of chromatin, right? So this is more about what happens to the chromatin rather than what happens to gene expression, although these will often be the same direction. And here's an example. So where, I would say for most TFs that are annotated as activators or as repressors, we can see this relationship, right, where positive or negative correlation, but there are also TFs that have unclear relationships. And here's this one on the right here that's a TF that is described both as an activator and as a repressor in the literature. But we, looking at least in our data, we see this negative correlation, which implies that it's likely acting primarily as a repressor. Okay, quick one-slide anecdotes. So one is, one of the novel findings from the atlases is that in addition to the spleen and the liver, we implicate the adrenal gland as a novel, albeit minor, site of erythropoiesis, and this is supported by a number of lines of evidence, including the detection of early erythroid progenitors in the primary data, as well as phagocytic macrophages in the adrenal gland, and then in situ validation. And in collaboration with Jim Pallas at Rochester, we were able to validate this finding in the mouse, showing it's not just specific to human development. GWAS signals, I think, is pertinent to some of the topics that we think about the intersection with human genetics. We can look for enrichment of cell type specific accessible chromatin at the intersection with heritability for particular traits. And we also saw this in our mouse chromatin accessibility, but here it's human versus human as opposed to human versus mouse. And I won't belabor the point, but I think that the key take home is there are rich opportunities here for implicating or nominating particular cell types as relevant to particular both disease and trait phenotypes that are available in summary statistic form through resources like the UK Biobank. We've, in a very preliminary way, started to look at kind of the equivalent of eQTLs, but at single cell resolution or single cell type resolution. And you can see a bit of that. So we have some anecdotal examples in the paper, but it's important to note that we only have 28 individuals, and many of which we don't have the same, sets of tissues. And so we're very underpowered to do this, right? And so there's really a big field or a big set of work ahead of us. We have the data is all available online at this website, atlas.bratman-beatty.org. Not only these data, but some of the other data from other kind of organism scale model atlases that we previously generated. And a lot of these data visualization stuff that we've been trying to work out has been the work of Michael Zager at the Data Visualization Center of the Fred Hutch and the Bratman-Beatty. And we're trying to really think ahead to not only how we look at gene expression interactively, but also chromatin accessibility. And then lastly, second to last slide, given kind of the topic of the session is kind of a transition back to any discussion we're going to have, I just thought I'd throw this up here. So the left is actually the paper in college that got me into genetics in the first place. And this is a very discrete phenotype, and you can see it's similar between the human and the mouse, where deletions of the c-KIT gene result in this piebald trait. And on the right is where my lab kind of got its start, which is in developing exome sequencing and applying it to Mendelian disorder. So this is Miller syndrome, where we were able to identify DHODH as the causative gene. I think the phenotypes here kind of give examples of what seems like it should be a relatively easier and a relatively harder set of developmental phenotypes to tackle with these kinds of single cell technologies. But I think even the easy example, when you really get down to brass tacks and think about, okay, how do we actually decipher the precise molecular events that lead to this phenotype, you realize even that supposedly easy example is going to be really hard, right? And so I think there's, just as maybe a transition point to the conversation, I think there are tremendous opportunities here at the intersection of rare disease genetics and these kinds of single cell atlases, but there are also big important challenges that we'll have to address. Okay, and I think I've acknowledged the key people along the way, so I'll stop there, and thanks for the attention.
Spectacular. Thank you so much, Jay and Heidi. Okay, so we've got questions specific to your talks. We've got also 3 very awesome panel members. I'd actually kind of like the panel members to just give some impressions. There's some aspects of what rare disease, this juxtaposition of rare disease and trying to get causal variants and figured out diagnostically versus what's the disease mechanism, what's the underlying biology mechanism, and then how are we going to translate that into really helping people and helping patients. So that is all part of the consideration matrix of Heather, Ewan, and David. So maybe if you 3 guys could start off a little bit of what are your thoughts in hearing this? How can Human Cell Atlas help the mission for rare diseases, and particularly given what Heidi and Jay have laid out?
So, I mean, I'll go first. The first thing to take a step back is we're still in this amazing kind of, discovery, sorting out of life kind of phase, and the Human Cell Atlas is sort of rebooting this fundamental space that we first had with microscopes, which was, a characterization of tissues by microscopy, and now we're doing a characterization of tissues using these omics technologies. And it's really hard to know and put a timeline on when things are going to come through. But I do think it is about working out in particular how particular genes function, and this increasing amount of pleiotropy of genes, so genes that do more than one function. And so for the rare disease community, what this means is that you will have variants in the same gene, very often or nearby the same gene, and they may give you different phenotypes. And to sort that out, you need to know kind of not only where does the gene mainly work, but also what of its side kind of tissues and projects. And some of you know this, that some genes do completely weird and wonderful moonlighting jobs somewhere else in the body that seem to have no relationship to their sort of main role in life.
And there's that contextual complexity too, of just that gene working in the context of other players that are expressed specifically with it. So its interactome is different in this context.
Yeah, and I think again, there's this kind of push of trying to connect up a particular gene with a particular piece of biology through these things like single cell atlases, but also really dissecting out a gene's function into its pleiotropic subcomponents. And very often those are tissue specific. It causes the rare disease by clinicians and biologists, maybe David can talk to this, quite a bit of a headache to try and understand why is it I get these 2 different phenotypes or 3 or 4. And then hopefully single cell data can help really narrow down to here are the different roles of this gene, some of which might be the same kind of process. And like I said, sometimes a gene just really moonlights somewhere else.
But I build on that. Is that fair? Right? I thought it was great that Jay gave as an example the paper that inspired him, which was the mutation of c-KIT in piebaldism, because that is the ideal example of tissues being made up of such heterogeneous cell types that no one would have suspected that a similar kind of phenotype could also be associated with megacolon or with other problems with deafness and so on. And so not only are we likely to discover a lot about the pleiotropy and the interaction of given genes within a specific cell, but we're also going to get a much finer grained view of the heterogeneity of different cell types around the body. And this discovery of new cell types in the adrenal gland, hematopoietic cell types, to me, it's a perfect example. And I'll be wanting to ask questions later about how fine grained we can get technically. I think this is likely to improve even more over the next few years, is how we're able to label and define cell states and cell types, and then find them in unexpected places doing unexpected roles, which is going to help explain some of these rarer conditions, sometimes even, you know, family or person specific conditions. So it's very exciting time.
Is there anything that the neural crest doesn't do? It doesn't help with? And what do we learn? And actually, I mean, that for Jay, did you guys see neural crest trajectories and, you know, going out different lineages? And was that there in the data, or were you a little too late for that in the human?
We were a little too late to see it going out. We absolutely see it in the mouse, right? Meaning in the earlier E9.5, E13.5, you see these beautiful, right, trajectories of, they're already separated to some degree. Yeah, but it's you're right. What does a neural crest not do? It seems. So.
And we have to remember that it's not just crest, of course, but what do cells not do talking to each other? So the future is in cell-cell interactions and knowing where they are in space and over time. And so combining all this information with the developing spatial technologies is just making us all so very excited. That's what we're trying to work on at the moment.
Sergiu Pasca's work, where he's doing organoids and then getting the organoids to interact with each other, or precursors of the organoids, and then getting sort of forced really A-B genetic interactions that can be tested in that context of cell-cell interaction. Really innovative stuff. David, do you have some thoughts on this?
Yeah, no, I do. I mean, I it's really fascinating watching the talks together, actually. So I think one of the things I'm most interested in what makes a cell vulnerable to something that looks like it should affect every cell in the same way. Things like, you know, cohesionopathies and, you know, DHODH is a good example. It should, you know, it should be affecting every cell that's going through S phase, really. But some cells are more vulnerable than others, and actually, if we can use some of the single cell data to try and work out what makes a cell vulnerable to perturbation. And particularly a lot of the disorders I deal with are really de novo mutations. So the parents don't have the mutation, the child has a mutation. But, you know, and a lot of them are what just look like general chromatinopathies, but they give very specific phenotypes. And that's a really interesting mechanistic way. I mean, I think your second point about how do you actually intervene with all this stuff, I think, until we know what the mechanisms are, I think that's incredibly difficult to work out how you would do that. I think
what's coming out about penetrance and allelic interaction and sort of genetic background effects for modifying allele severity?
Yeah, I mean, I think I'm probably the worst person in the panel to ask about that, because most of the disorders I deal with are really catastrophic, you know, single base changes that are catastrophic to development. And actually there's very little influence of modifiers there. So I think, whereas I think if you go later into, you know, adult onset disease and maybe even later childhood, you'd have a different view of that. But a lot of the conditions I deal with, the actual modification is mostly in the severity of the mutation, I think.
Heidi, where are you at in that business?
Yeah, I mean, it's interesting. Some of the genes we've been discovering or collaborating and discovering in the neurodevelopmental space have been challenging because we might have a set of, let's say, 8 to 10 cases with de novo variants, highly suspicious, for a particular gene. But if you look across the phenotypes, there's variation. There may be broader neurodevelopmental phenotypes. Some have epilepsy, some have seizures, some have, learning delays. They're sort of on a spectrum of related phenotypes, but not perfect matches. Not like, oh, everyone with this disorder has retinitis pigmentosa and hearing loss, where it's clearly they're all the same. And I think, that's undoubtedly, sometimes we get enough cases where like, we think this is absolutely the cause, but why is there such variability in these individual phenotypes that are manifesting in different ways? And I think they undoubtedly have to do with some sort of both genetic, environmental, or even stochastic, modifier effects. But those are things that we struggle with when we're trying to say, have we found the cause, or is this just a collection of spurious, unrelated de novo variants in the same gene? And I think we struggle a little bit. The other area that we struggle with right now is, although we used to think about germline disorders and somatic cancer as like the two, different ends of variation rising. Now we recognize more variation that occurs early in development, but after, before embryogenesis, and where you have effects that are in a subset of tissues, but not the entire constitutive set of cells. And that undoubtedly will, overgrowth syndrome is a great example, but there are more and more examples of disorders that might arise in tissue in some stage of development and then manifest in different ways, depending on which tissue and cell types have been affected in that body. And that I think can lead to a lot of variation in phenotype as well. And of course, when you take blood samples from these families, you don't find it because you haven't actually looked at the right tissue.
There's a question from the Q&A about the role of epigenetics in as a causal factor, and maybe exposures of the mother and child in utero to stresses that could cause epigenetic effects. Do you see that as
Bruce, could I just
driving?
ask a very simple question, kind of before we move on to the epigenetics, which is just simply, what's the evidence that expression levels matter in order to affect cell types or lineages or compartments and so on? I mean, particularly, I guess, David and Heidi, but maybe also Jay or others here.
I mean, we certainly have lots of great examples of where the mutation and the severity of it is correlated with phenotype. So it really demonstrates that the dosage effect of a particular variant is important in thinking about that. So
sarcomeric proteins.
Yeah, absolutely. LDLR is a great example, of dosage effect, but it's a simplified one.
Anything that's haplosufficient, right? That's the presumed. It's presumed. It's not certainly the case, but it's presumed to be a dose effect, right? But one can think of more creative possibilities.
Absolutely, yeah.
But I meant that in terms of so I meant also in terms of understanding where the mutation is acting. Basically, that you can just kind of not just, but that you can that the expression level in the reference is kind of predictive of where the.
I think it's a great point, Sarah. Like that's not a proven. That's not like it's hard to think of killer examples, right? And probably the killer examples one can come up with are not the ones that are going to generalize, right?
Well, an example sorry, go ahead.
Go ahead.
Well, example I was just working on this morning. Family contacted me about their cadherin 23 mutation. And some of these mutations lead to Usher syndrome with hearing loss and retinal defects, and others are just isolated hearing loss. Well, it's become clear now that the reason for that is the milder mutations that are only missense mutations and that have mild effects lead to the isolated hearing loss, whereas the more loss of function mutations lead to retinal disease. So they're not domain specific. It really seems to be a threshold effect that somehow the hearing organ is more susceptible and the retina more resilient. And that's why you see these different phenotypes depending on the severity of the mutation.
Are there
And
expression level differences in those two compartments, or do you think that there's complementing cadherins that could make up for the hit on the one?
It's a great question. And, across the different Usher syndrome genes, several of them have this effect, and some of them are transcript specific, where one transcript is expressed in both tissues and another transcript is expressed in just one. And we can see that all the mutations for Usher are in this region.
Alternate splicing. Oh
Yes,
my God.
but
So
the
single
cadherin
cell's not
23,
getting there as fast as we need to.
yeah, exactly.
But I mean, I think one of the interesting things is where you've got bilateral organs like the ear and the eye. Actually, there are some very, things like the SOX2 mutations where they seem to be just straightforward haploinsufficiency. Most people have got 50% of SOX2 will have bilateral anophthalmia, but a proportion of them will have unilateral anophthalmia, and the other eye will be completely normal. So within an organism. So I think Heidi's right. I think stochastic effects, which but presumably, one of the things about single cell data is it'll be able to identify some of these very critical bottleneck events where you can actually go one way or the other. But I think the bilateral organs, I think, are an interesting group to think.
Is there evidence that environmental effects at a particular developmental stage can translate to genes that are on the edge of severity penetrance?
I mean, there's definitely, Heidi, go for it.
Yeah, I mean, some of the work that on like Titan and dilated cardiomyopathy, for example, that James Wares has been doing, showing alcohol, impacts of the expression of dilated, of sarcomere, or well, not sarcomere, but dilated cardiomyopathy mutations in Titan, where with the alcohol exposure, it pushes you over the edge. That's one example that comes to mind.
I think there are other examples. There's that very nice lung, it's not alpha trypsin, but there's a lung rare mutation which is exacerbate where the interactions with the environment, but also the other, I think, big interactions that we're seeing is common variants with rare variants as well. And so that's often in some sense sometimes in the same gene. So this is like it's sort of extending the concept of a complex heterozygote into a new space of weak penetrance. And again, I think it's just, we have a lot to help sort out, and rare disease and common disease are coming closer and closer together, and one has to treat it more as a continuum. And that goes to again, just to bring this back to the Human Cell Atlas, it goes to needing a kind of fundamental understanding of human biology, tissue biology, and then physiology to interpret what's going on. And we don't have to know all the answers to give really good advice to children and parents, or to find drugs and everything else. So I think it's also important to realize that every time we make more, we gain abilities in doing this diagnosis or separating out why we get these different complex phenotypes. We're going to improve things without necessarily coming up with a complete answer to everything. All of this will be incredibly useful, but it's enabled by having these large reference data sets, be it the genome, be it SNPs, be it Nomad, or be it the Human Cell Atlas. So these large reference data sets that the whole of the world can use and don't have to regenerate, every time they have a question. Rather, it is a more like a sophisticated lookup in the right way. It's one of the reasons why I'm so keen on the sidelines with the Human Cell Atlas. So keep going, Sarah. That's
everybody else.
And everybody else, yeah.
All over the world.
We're adding more dimensions all the time, right? We've just had, Jay mentioned time, right?
Yes, yeah. I think
we're introducing more and more dimensionality.
Yeah, and we shouldn't be scared of that, but one has to treat it with respect and that
Humility.
yeah, you know, it just you know, some of those dimensions, again, it goes to what we mean. What do we really mean when we say there's stochastic effects? And I think David's very right to pick up on bilateral organs as a good example where you know, we know that very many of these processes must be largely identical left to right, though it's, you know, don't be tricked by the symmetry of our faces. You know, our bodies are not naturally symmetric. The face symmetry is a highly processed and manipulated thing to get us to look symmetric. But that's true in developmental disease. It's also very interesting, I think, in cancer. So it's not the case that if you have kidney cancer, or if you have breast cancer, you know, the odds ratio of the other kidney or the other breast having of catching cancer in the other organ is higher, but it's not massively higher. I mean, it's not, you know, remarkably higher. And that tells you that there's a lot of stochastic behavior that leads to disease, be it developmental or be it, you know, late onset.
What are the stochastic mechanisms? Is this epigenetic effects? Is it cell replication, extra apoptosis?
Bruce, every time anybody says the word epigenetics to me, I would like to come back and say, now, just to be clear, do we mean somatic cell state epigenetics, or do we mean transgenerational epigenetics? Because I think you know, and we could do parent of origin epigenetics as well as like a third bonus type of epigenetics.
I was thinking more flip states of you know, alternate differentiation ratios, or you know, just the emergence of a daughter cell type that is maybe a hard event, and then here you impair it with a mutation, and then you know, fail to develop the structure properly.
Yeah. Doesn't this come to the heart of what a dominant and recessive gene are? So I mean, basically, it just means is 50% enough? And if 51% is enough, and you then it's recessive and 49's not enough and it's dominant. I mean, I think, but that's not how life works. And actually, if you've got variability in that, you know, you've got a noise in the system, at a critical point during development, you can imagine that just if you're on that boundary, just random events, true stochastic events will change fate. So that's the way, at least for some of the disorders, I think that's.
And exon activation is actually a great example of, you know, a largely stochastic process in terms of which cells are inactivated.
But I do think that quite a lot of these stochastic processes, you know, exon activation really does feel like somebody's kind of cosmically flipping coins somewhere in exist or whatever. And there's these very random things. I think, you know, I think there's a very broad range of stochastic events which are really kind of micro fluctuations in exposures and things like that, which are so, you know, they're so impossible to track and know that we end up, the only way we can talk about them is that they're stochastic, even though formally they in some sense they're, oh yes, the virus, you know, you breathed in and your right lung got a little bit more of it than your left lung, and if your left lung is your right lung is a bit more exposed. You know, in some sense that's a predictable event by how you breathed in at that particular moment of time. But it would be kind of crazy for us to try and measure all of those variables and all of that. I mean, utterly impossible. So there is this sort of, you know, the way we use the phrase stochastic event, I think encompasses everything literally from cosmic rays hitting atoms in chromatin near exist that switch exon activation from one haplotype to another, but all the way up into this much more kind of something random is going on which we can't track, basically. Yeah.
Is there anything clear from gender identity development? How is that epigenetics? Is it?
Wow, Bruce, you're really
underlying genetics? I mean, is that going to be a good example?
We want to go into this particular rabbit hole. It's quite a rabbit hole.
Let me give a entirely different example. Nori disease was the first disease I studied as a PhD student. There's a degenerative hearing loss. And you might think it's just sort of just general degenerative process, but if you actually test and ask the patients, they have normal hearing and then some event happens and they lose their hearing overnight. And then it comes back, but always at a slightly less. So there's these events that happen and they talk about, oh, I had a really salty meal and actually, and the next day I lost my hearing, or weird things. And it turns out we actually know that the underlying effect is through a vascular defect. And so it's very possible that the vascular dynamics that are going to be fluid throughout the body are actually contributing to this sort of, very sort of episodic loss of hearing as opposed to a gradual, accumulation of, material or something. So I think those are just examples of where the dynamics of our human body can have, stochastic effects.
I mean, one of the things which I love, by the way, about Jay's work is that we are not only like myopically focusing on human, obviously mice give us all this accessibility, but I love the Drosophila and, C. elegans work as well, because those are organisms which we can just cover, we can kind of know in such depth, Jay. And I'm right in saying that your single cell work recapitulates the Solston lineage tree, as it were. Can you reconstruct
it? So I mean, if you get a chance, I mean, we were not involved with the follow-up on this, but Packer et al. Science 2019. This is from Bob Watterson and Cole and others. It's an amazing paper where they really do a time series and integrate it with the Solston lineage, and it's just remarkable. I do.
Jay, there's a highly voted on question about your work that with regards to how far do you think you really got in terms on the annotation of the different lineages in the paper? How much more work could be done when people really dig into a particular area, stage, tissue, group of cells? How deep is are some of these differences between cells that are the basis of taking trajectories or the states that they're in? What do you think the level of mistakes are?
Well, let me. I just want to finish one point about the model organism. But I do think, I mean, just touching across the entire discussion we've had so far, it is very difficult for me to see how we make any headway on any of these questions about developmental genetics intersecting with single cell data without focusing on model organisms and early development, right? You can't access the time points you need. You can't access the numbers you need to be able to really recapitulate a process over and over and really understand what's going on. So, I think that the human parts are great and important, but really to answer almost any of the questions we've asked so far about this interplay and how developmental genetics really plays out to give rise to pleiotropic and complex phenotypes, there has to be some really narrowing in on good models for asking those questions. I just want to make that point, which I think speaks to something Ewan was bringing up. And then I guess to get back to your question, Bruce, right? It is like, it's this challenge of this rabbit hole of how far you go down, like endless annotation, right? And we certainly didn't do as good a job as we could have done, and I think we really relied on existing atlases to, in transferring, particularly for the subtypes. And there's a point beyond which you're just naming subsets by markers, and without additional information, be it spatial atlases or whatever, it's going to be hard to really to annotate those meaningfully. I do really like this idea of can one build a classifier to separate these two clusters, really robustly and well, and as a means of trying to decide when subclustering has gone too far, right? I do think that's kind of hopefully a useful tactic, right? Because it can just seem to go on forever. And the other,
how large were some of those mouse-human differences, do you think? Are there
I was amazed how much it all just kind of lined up. I mean, part of the challenge was that we were looking at two different windows of development. And so we couldn't tell whether differences were due to time or species. But now having done similar exercises with quite other species like frog and zebrafish and trying to bring them in, you realize just how close mouse is to human and just how different these other things are, right?
We're running out of time.
Oh, I'm sorry.
There's several other questions having to do with nomad. Heather, do you want to bring up your point that was well voted on by the crowd in terms of?
I mean, some of the questions were answered in text. So
I was answering away a bunch of questions.
yeah, Heidi answered some, and I can wait on others.
Some of the things we're dissecting in single cell are different gene non-coding RNAs. We're actually going to have a cool talk from Piero Carnizzi in the meeting the Asian meeting coming up on that, the role of non-coding elements in regulating development. Heidi, are you seeing those kinds of genes scoring in terms of causal effects? Are some of their effects so strong that those embryos don't survive? What do you think is going on in kind of non-coding space for disease roles?
Yeah, this is a tough nut to crack. We are starting to look at non-coding space, trying to understand what the lots of variation that's out there and whether any has impact. And as we build Nomad into a larger and larger resource, we become more powered to be able to look at constraint, not only regionally within genes. So sometimes a gene may not appear constrained, but if you look at one end of the gene, that region might be constrained. So members of the team, Catherine Chow, and working with Conrad and Caitlin Smocha, are developing algorithms to look regionally across genes, but also the same approach is starting to look at non-coding region so that we can start to look at constraint, which will help us prioritize variation in non-coding region for functional studies, because there's just too much of it right now to be able to identify what to focus on. And RNA-seq is also quite useful for deeper intronic variation that's hard to find signal out of noise, but when combined with RNA-seq data, you can start to zero in on genes that have transcript alteration and therefore identify potential impacts. So I don't know if that quite gets at your question.
It's an incredibly important point that you brought up. Just the significance of looking at intolerance of variation and as a primary look at a gene region, and so is that factored into the presence of a mutation well enough by current classification algorithms? And how do we best see that?
Yeah, I mean, most of it's been used today for gene level prioritization. There's a paper from Nikki Whiffin's group using this regional constraint approach for, I think it was MEC2F. I can't remember exactly. It just was posted on bioRxives, using this approach, and you can see this, really high correlation of constraint at one end of the gene, which is correlated with all the pathogenic variation. So I think we're going to be starting to use these approaches to help identify target regions that are correlated with disease that are harder to bring out if you're only looking at the whole gene level.
Thank you so much, everyone, for this fantastic session. This was really a treat, and it's going to be fun to watch again in slow motion. So bye-bye, everyone, and thanks for joining us.
Sure, and just some closing notes before everybody leaves. So again, thank you for joining us for the Human Cell Atlas Developmental Clinical and Rare Disease Applications Seminar. We definitely want to thank the developmental program committees. So Bruce, who hosted today, Gary Bader, Elaine Chedidal, Dana Pear, Muslifa Hanifa, Sten Lenarson, Aviv Regev, Deanne Taylor, and Sarah Teichman. And we also want to give a very big thanks to the HCA Executive Office and Scott and Russell from the Broads video production team. We want to thank Heidi.
And to you, Christine, so much. Thank you.
Thanks, Bruce. A big thank you to Heidi and Jay for amazing talks. I think our audience really appreciated it, and I know you were able to follow up with people on Slido, so thanks so much, and I'm sure they appreciate that. Thank you, Heather, Ewan, and David for joining a wonderful panel discussion. It certainly seemed very intriguing and awesome to my non-scientific ears. Just want to point out that we do have an Asia and Australia Clinical and Rare Disease Applications seminar that will be hosted by Bruce Aronow, and that will take place December 16th of this year, 7 to 8:30 a.m. GMT. The speakers and panelists for that will be Daniel MacArthur, Piero Carnizzi, Jay Shin, Heather Etchevers, and Chrissy Lim. So if you're awake in the States, feel free to join us for that one. And please encourage your Australian and Asian collaborators to join us for this. The next developmental seminar will be on computational modeling. We are aiming to schedule the Americas, Europe, and Africa edition the end of January. So please stay tuned for dates. Two of our confirmed speakers and panelists are Dana Pear and Alexander von Udernaden. So I'm sure that will be a great discussion to tune into. And we will also have an Asia and Australia edition, which we hope to schedule at the beginning of February. And two of our confirmed speakers are Shyam and Guoji Guo. So please join us for that. As always, please visit our website for more information on the seminar series. And if you are not a project registry member, please join us. And Tracy should have linked that into the chat. Just so you are aware, recordings from today's seminar will be posted on YouTube and our availability site. So please check there. And please fill out our survey on Slido. We want to hear from you, and we are always looking to continuously improve the seminar and all HC events. So please take the time to fill that survey out. Great. So just to let you know, the HCA Biological Network Seminar Series kickoff meeting will be January 14th, and that will take place 10:30 to 12 p.m. The two featured networks will be the Breast Network and the Liver Network. And just to let you know, the HCA General Meeting is going virtual this year. It will take place June 7th through June 9th, 2021. So please be on the lookout for save the date information, which will come soon. And the HCA Developmental and Pediatric Cell Atlas meeting that was supposed to happen in Toronto of July this year is going to be postponed. We're going to bring that meeting virtual this year, and we're looking to reschedule to August 2021. So again, please stay tuned for more information. And thank you again for all of your attention, and have a great day.